I open this academic ceremony in which Nikolas Rapp will defend the academic thesis Chasing the Perfect Storm, Vascular Calcification in Chronic Renocardiac Syndrome. May I invite you to present a summary of your study and the main conclusions of your thesis.
Highly esteemed Pro-Rector, members of the Corona, supervisory team, friends and colleagues present here and online, welcome to the defense of my thesis. Thank you all for being here today. In the next 15 minutes, I will show you where a storm is brewing in a chronic renocardiac syndrome. To introduce you to the topic, I will walk you through the title of my thesis, and I will do so starting from the back. With the term chronic renocardiac syndrome, which is defined as chronic abnormalities in renal function leading to cardiovascular disease. That's quite a mouthful, and you probably have never heard of that. So why did we study exactly that? Well, the short answer would be because it has a big impact on people and society. And I just brought a few statements to show you what it means. And it means that across all ages, the expected remaining lifetime for people with advanced chronic kidney disease is more than halved. It also means the average 5-year survival of patients starting dialysis is lower than that of cancer upon diagnosis, and the costs are higher. And the majority of chronic kidney disease patients will die, and mostly from cardiovascular disease, before they reach the stage of dialysis or transplantation. And for the 500 million affected people globally, that means 1 million premature deaths every year. But how does it come that far? Well, under normal circumstances, a healthy kidney, among other functions, filters the blood, maintains mineral homeostasis, and regulates neurohormonal processes, while the cardiovascular system, with elastic arteries, maintains normal blood pressure, organ perfusion, and blood clotting. Both systems work closely together and regulate each other, to maintain an harmonious entity. If, however, one of the two malfunctions, it has negative effects on the respective other system. If a person first develops chronic kidney disease, it will subsequently lead to damage to the cardiovascular system. Chronic kidney disease leads to an impaired blood filtration, as well as mineral imbalance and hormonal dysregulation, which negatively affect the cardiovascular system. This will lead to a destructive feedback loop, where the damaged cardiovascular system, in turn, leads to even more severe kidney disease. This effect of chronic kidney disease leading to cardiovascular disease, which then enters a detrimental feedback loop, is called the chronic renocardiac syndrome. Effects of chronic kidney disease on the cardiovascular system can be heart failure, hypertension, and vascular calcification. Vascular calcification is also the second part of my title, and it refers to the undesired buildup of mineral within the vascular wall. And everybody can deposit mineral in their arteries, and in fact, most people do so by the age of 70. This can happen all over the vascular tree, from the heart valves to the aorta, and other vessels like the larger vessels in the upper leg, and all the way down to small arteries in the feet. Larger arteries, like the aorta, consist of 3 distinct layers, which are from the outside to the inside, the tunica externa, tunica media, and tunica intima. The tunica media is the most prominent layer and contains vascular smooth muscle cells, which are not only the main cell type present in larger arteries, but are also key players in the development of vascular calcification. It follows that vascular calcification can occur in the tunica media, where it is referred to as monkeyback sclerosis. Or it can occur in the tunica intima, where it is associated with the well-known fatty plaques of atherosclerosis. But why is it we worry so much about vascular calcification? Well, research has shown that vascular calcification leads to detrimental comorbidities like hypertension, and subsequently also contributes strongly to the 1 million early deaths annually related to the chronic renocardiac syndrome. And while most people will develop some degree of vascular calcification, chronic kidney disease patients are at the center of a unique set of risk factors, the combination of which is sometimes referred to as the perfect storm for vascular calcification. To grasp what's so stormy about it, one needs to understand that calcification is not per se bad. In fact, in some parts of the body, it is desired, for example, to maintain healthy bones and teeth, while in other parts, like the vasculature, it is not appreciated. It follows that in the body there are calcification promoters and inhibitors, helping to tip the scale into the desired direction in each compartment. In the healthy vasculature, the inhibitors outweigh the promoters, and the scale is tilted towards non-calcifying. If, however, the promoters gain more weight or the inhibitors are lost, the scale might tip in the wrong direction. And to some extent, that happens to most people while they age. But in chronic kidney disease specifically, a lot of calcification promoter clouds cast their shadow on the cardiovascular system. And now the presentation is stuck. There it is. And there are the clouds. And yes, in CKD, a high pressure of factors that drive vascular calcification, like inflammation, mineral bone disorder, and uremic retention molecules, and a lack of factors that inhibit vascular calcification, like vitamin K. And this set of risk factors, all coming together in CKD, is unique and sometimes referred to as the perfect storm for vascular calcification. And now it is stuck again. There it is. Exactly. And to study this phenomenon, we tried to capture that storm in a dish. And for this, we developed a test system, which we called the biohybrid. In principle, we combine a fraction of the blood, called serum, with vascular smooth muscle cells, add a stimulus to support calcification development, like a little bit of extra calcium, and lastly, add a fluorescently labeled protein called factorin A. This probe will bind to calcification if it develops over time, which will lead to a bright orange signal. This means a stronger calcification pressure in the serum will lead to a stronger response in form of more signal in the biohybrid. This signal we can pick up by taking and analyzing images, and the stronger the calcification pressure in the patient's serum, the more orange signal we will see. This signal we want to combine with other information of the patient, like its medical history, standard laboratory tests, and thereby contribute to better patient treatment decisions. And the application of the biohybrid is twofold. It may be used to detect serum calcification propensity, but it also can be used simply to detect vascular calcification in any experiment. And this has several advantages. As in vitro calcification detection system, the biohybrid has been shown to be more sensitive compared to the test that is currently used routinely to detect calcium in vitro. It additionally needs less serum sample, and it allows the researcher to follow the process of calcification over time, allowing for a less subjective readout. In the next step, we applied the biohybrid to serum of patients and tested the calcification propensity of serum samples of several different patient groups. Firstly, we used serum samples of patients with a good kidney function and compared it to serum samples of patients with poor kidney function. In the biohybrid, the response was much stronger with the serum of patients with poor kidney function, and this corresponds to what we see in the clinical situation, where chronic kidney disease patients show much more vascular calcification. Secondly, we tested serum of patients with very late-stage chronic kidney disease, which receive dialysis treatment to maintain their blood. Dialysis is supposed to filter the blood when the kidney can no longer fulfill that function. And in this case, we looked at serum collected before and after dialysis of the same patient. And the biohybrid reacted a lot stronger to serum taken before dialysis. And this again confirms what we see in the clinic, where dialysis is also used to reduce calcification pressure. We also looked at patients with normal kidney function that still develop vascular calcification. And in the biohybrid, the serum of patients that already show a lot of vascular calcification reacted stronger than the serum of patients that had little or no vascular calcification. This indicates we might be able to use the biohybrid to determine the calcification status of a patient. Lastly, we used serum of patients with normal kidney function that either received a certain medication called warfarin or patients that did not receive this medication. Warfarin is an antagonist of vitamin K, which itself is a potent calcification inhibitor, and it is known that patients that receive warfarin have higher risk of developing vascular calcification. And this is also what is reflected in the biohybrid response, where serum of patients that receive warfarin reacts stronger compared to serum of patients that do not receive it. Next, we further developed the biohybrid system. The biohybrid test requires cells as biosensors to react to the serum and act with the calcification response. What we had done so far was using cells which we directly received from patients called primary cells. These patients underwent heart or aortic surgery, and sometimes the surgeon must remove tissue to repair a defect. The patients can donate this tissue, of which we can isolate cells to work with. Now our lab has established a different method to obtain cells via induced pluripotent stem cells. We can now use a tube of blood, isolate cells from the blood, and use these cells to create stem cells. The stem cells, in turn, can be used to make many different types of cells, also the vascular smooth muscle cells that we need for the biohybrid. This method provides a more secure source of cells, and it allows us more flexibility in the development of the biohybrid. Additionally, the use of these new cells are also led to better repeatability of our experiments. And with this, we were able to further expand the spectrum of patient serum we tested. Additionally, we tested the biohybrid as a platform for medication screening to see which medications can potentially be used in the treatment of vascular calcification. Lastly, we also applied the biohybrid and tried to predict vascular calcification present in patients from their blood. Now, the serum that we've used so far is a very complex mixture of many different components. In the next project, we aim to identify which serum components actually contribute to the development of vascular calcification in the special high-risk population of chronic kidney disease patients. And for that, we looked at a group of molecules which are specific to chronic kidney disease patients called uremic retention molecules. These are waste products produced or taken up by the body, which due to failing kidney function cannot be properly excreted into the urine anymore. They are commonly regarded to be toxic and responsible for many detrimental effects in chronic kidney disease. About 150 different substances have been classified as URM. Which of those are directly involved in vascular calcification development was not very clear when we started. So we reviewed the literature and found, actually we didn't find much. Only very few of the uremic retention molecules have been linked directly to vascular calcification, leaving a huge knowledge gap. But we found information linking the uremic retention molecules to processes that precede vascular calcification. So we used that information of the processes that precede vascular calcification to rank the URM according to which ones are most likely to induce vascular calcification. 4 of the top-ranking URM were then selected for experimental testing. And 2 of those 4 did indeed increase vascular calcification in our cell-based test system. One of those, called paracrysyl sulfate, is one of the most toxic URM and known to have many negative effects on different organ systems, and has been termed prototype uremic retention molecule. It was then chosen for an in-depth experimental analysis. And in summary, we were the first ones to show its procalcific effects in vitro. Furthermore, we contributed to unravel the mechanisms underlying its procalcific effects, and additionally, we could show that vitamin K can counteract the procalcific effects of paracrysyl sulfate. In our last experimental part, vitamin K was also the main objective of our study. We looked at the vitamin K status of 4 different patient groups, all of which have a high cardiovascular risk. Vitamin K is utilized in various tissues, including the vasculature, where it is a potent vascular calcification inhibitor, and the liver, where it plays an important role in a protein modification step called glutamic acid carboxylation. If there's not enough vitamin K, it leads to unfinished proteins which lack this modification. And in the liver, this leads, for example, to production of protein induced by vitamin K absence 2 or PIVKA2. And in the vasculature, this leads to UCMGP. And because vitamin K is notoriously difficult to measure, these proteins are often used as so-called surrogate markers to measure vitamin K status. And this is also what we did. And we looked at 4 different patient groups, 2 with chronic kidney disease and 2 without. And we found that patients with normal kidney function, but which do have vascular calcification, appear to have normal vitamin K status. And we also found that PIVKA2 and UCMGP, despite both being vitamin K dependent, only correlate very poorly with each other. Furthermore, we found that warfarin therapy, which is a vitamin K antagonist, increases UCMGP. And conversely, vitamin K supplementation leads to improved vascular vitamin K status in form of better MGP carboxylation. In summary, the key finding of my thesis are that we developed the biohybrid system, which is a novel vascular smooth muscle cell-based test to detect calcification in vitro and to assess serum calcification propensity. And subsequently, we improved the biohybrid system using stem cell-derived vascular smooth muscle cells, expanded the patient basis, and explored its application in drug screening. Thereafter, we investigated the role of uremic retention molecules in vascular calcification and found the impact of uremic retention molecules on vascular smooth muscle cell calcification remains largely unknown. And we concluded better insights into their effects is key to develop strategies to reduce vascular calcification. We then took a closer look at one particular uremic retention molecule, paracrysyl sulfate, and found it exacerbates vascular calcification in vitro, and that vitamin K counteracts this detrimental effect. Lastly, we investigated the vitamin K status of 4 different patient groups, all of which have high cardiovascular risk. And we found that vitamin K status appears to be normal in patients with vascular calcification and normal kidney function. And additionally, both vitamin K-dependent proteins only correlate very poorly with each other. So I hope this wasn't too much of a storm of information, but just the perfect amount. And hereby, I'd like to hand the work back to the prorector.
Thank you very much. Well, it was a lot of information, but it was very clear. Thank you. It's now time to start with the opposition. And the opposition will be opened by the chair of the assessment committee, Professor Delhaas. He's professor of biomedical engineering at Maastricht University.
Thank you, Pro-Rector. Dear candidate, congratulations on your thesis. I also would like to congratulate you on the cover. It reminds me of my days in San Diego, living there for 1 year and chasing the perfect wave, although I was really stupid by taking not a longboard, but starting to surf on an eggboard of only 6 feet. So I hope you do better. Then coming back then to your thesis, I was a little bit puzzled on page 63. Maybe you can open it and then have a look at the first sentence of this page.
Yes.
It states uremia in Greek literally means urine in the blood. Can you defend that sentence?
Yes.
Because I won't agree, but let's wait and see what you will tell me.
All right. Firstly, thank you for taking the time to assessing my thesis and also for chairing this committee. I much appreciate it. And indeed, the 6-foot egg is really challenging for a beginner. I bought a longboard. Yes, uremia in the blood, urine in the blood. Yes, it refers to, what I meant with this was that it refers to that in very late stage chronic kidney disease, patients don't excrete the uremic retention molecules anymore, which would usually form the blood. The urine, and not only the uremic retention molecules, but they also retain all
but you have to focus on what you really state there, that it literally means urine in the blood. I don't think so.
But I'd say, well, the urine that is usually excreted then just remains in the blood.
That might be true, but the word itself is, I think, it's referring to urea, which is the main component of urine, but not the urine itself. And that brings me to, what do you think, why those molecules, those uremic retention molecules, cannot be excreted? You talk somewhere on page 130, thank you, on the size of the membrane, but is that the reason? In real life,
the size of the membrane.
Is it the sizes of the pores in the membrane that determine whether you can excrete, those molecules, or is it other mechanisms?
Well, the size of the membranes, I guess, refers to the
pores of the membrane.
The pores of the membrane refers to the dialysis membrane of a dialysis machine, while the kidney itself, I guess, has a different functionality.
Yes.
Yeah.
So how comes then that if you have hemodialysis, you cannot get rid of those uremic retention molecules, not that good as you would be able to do in normal life? That's my main question. Well, I think it doesn't have to do so much with the membrane pores. It has to do with the fact that it's not a passive process in the kidney. It's not purely a filtration that is driven by pressure, but there is also active processes by transporter molecules across the membrane. But this is only, it's not my field, but I was struck by this being trained as an MD as well. Maybe we can go to chapter 4. And then that is in chapter 4, you develop a score, yes. Or is it page 63, I think? No, it's not. No, page 92, it's chapter 5.
Yes.
There you say you develop a score, and then, yesterday evening I tried to read the table, which I could not do because of the font size. But now I can do, and then still I have the problems that I cannot find those 4 uremic retention molecules in the table that you are saying that you are going to test.
That I'm going to test? Well, paracrysyl sulfate is the first one that is not red, and it's the first 4 that are not red, except for the resistin, and that's because the resistin is actually a protein, and because this was a trial and the proteins to buy them is much more expensive than small molecules, we decided to use the cheap ones.
Now I do understand what I did wrong, and that has to do also with the font size, that it is in red, it's labeled the ones that are described, and then the white ones are the first high scores that are not described. Okay, now I got it.
Exactly.
Now, then thank you for this. then I would like to go to figure 2, which in combination with the panels A and C, or A and B, strike me a little bit. In panel A, you have on the y-axis a u-gram of calcium per u-gram of protein.
Yes.
And then you talk about the effect of paracresyl sulfate, which is the pre-final column.
Yes.
And that is a concentration, according to your line 5, that's a concentration of 1 millimolar.
Yes.
Tested. Now, if you go then to the right, to panel B, and you look at the height of the column of the 1 millimolar paracresyl, that is clearly different from the one in panel A. And the same holds for the acrolein column in panel C. The comparable concentrations, they have different heights. How comes?
That's just variability in different experiments. The general size, so the effect size is not always the same. And what we look at is the relative effect size. So also the controls sometimes calcify more, calcify less. And that's why we look at relative effect sizes instead of absolute effect sizes.
Okay, and they can differ, a factor of 2. No, it's just, how far can they differ? Because that means that the sensitivity or the specificity of those tests are really a cumbersome thing.
Yes, they are. Working with cells, there's a high inherent variability in those experiments. Okay.
And that can you, in that sense, then also explain the results on page 47? Because on page 47, what struck me was that it might be advantageous to take rivaroxaban.
Yes, it appears so.
And why? Is that because that is, the test giving a result which is not predictable? Or is it because it's really advantageous? Well, or is it because it's a multiple choice question? Or is it because it's not a standalone effect of rivaroxaban? How do I have to interpret this result?
Well, whether or not it's a standalone effect of rivaroxaban, I cannot tell. Nobody has, in my experience, ever looked at rivaroxaban directly on the effect of vascular calcification. It has some effects on the parts which are linked to, so not rivaroxaban, but rivaroxaban's target. So activated factor 10 has been linked to some processes that are also precede vascular calcification, increased migration, for example, which then hints towards a de-differentiation. But whether this is a systemic effect of rivaroxaban or not, I cannot tell.
Okay.
But again, the relative difference in these experiments may indicate that it might be beneficial to take rivaroxaban compared to Markham-R.
Yeah. Okay, then taking your answers into account, I then think that you will tone down your conclusion on page 84. That you say that you show that small molecules in CKD serum significantly contribute to VC, because you did not prove that they contribute. You only have your assay that shows that there might be an increase in the assay of vascular calcification relation, but not in real life. And I would like to thank you for your answers, because they clearly cleared up many aspects of your thesis.
Thank you.
Thank you very much, Professor Delhaas. The opposition will be continued by Professor Stoll. She was also a member of the assessment committee. She's professor of genetic epidemiology and statistical genetics at Maastricht University.
Thank you, Pro-Rector. First of all, let me congratulate on your thesis and also on the very clear presentation. I'm a geneticist. I know nothing about that topic, and we will see whether it gets you in trouble or me in this defense. I actually would like to pick up where Professor Delhaas ended his question. And that relates to your proposition 3. Maybe one of your paranymphs could read that.
Human iPSC-derived vascular smooth muscle cells are superior to primary vascular smooth muscle cells for disease screening and drug testing.
Okay. So considering the remarks by my predecessor, why? Why are iPS cells superior to the normal smooth muscle cells in your hybrid SA1? Because when you think of iPS cells, they're redifferentiated from pluripotent stem cells, and usually they're from their biological state more juvenile. So do you have an answer for that? Or could you explain why you made that statement?
Yes, I can. Highly esteemed opponent, thank you very much for the question, and also traveling all the way here from Münster. Yes, so firstly, the IVSMCs, they're in a more juvenile state, that is true, but we actually consider it an advantage, because when we think about what calcification develops in the body, it also does not happen to a 70-year-old just very suddenly that already has seen a lifetime of negative influences like smoking and dyslipidemia and such, which are already present in the vascular wall, but it happens already a lot earlier, so we can see vascular calcification by the age of 25. So we know that also juvenile cells are affected by these factors, and when we really want to go to the mechanisms where it started, I think having juvenile cells is a lot more perhaps meaningful than looking at cells that when we get them, we work together with the surgical department, and then we get them from surgeries from people that are usually 65 plus, and also when they undergo heart or aortic surgery, they're not healthy, which means also the tissue that is removed is not healthy. So we don't actually know clearly that the effects that we see are either linked to the predisposition to what they've seen before, or the arteries have seen before, or if it is the effect that we have. So we would need a much larger N to rule out this, and with the IVSMCs, we get more of a clean model to work with.
Okay, let me jump into the more clean model of the iPSC cells. At least in my hands, iPSC cells are quite variable. And it's depending on the cell line and the correlation between moon, stars, and sunshine, how they work in the cell culture. So what's your experience with the variability of your system?
That's a very good question, and if I could, I'd forward it to my paranymph Cengiz, because he's actually the one that works with the iPSCs, and I only get the cells when they're already developed. But what I know from what he has told me from working in the lab is that the iPSCs are indeed very difficult to work with. And it's not only the iPSCs which vary, but it's also once you have developed the iPSCs and you go into different differentiations, we see differences between those. So we do see batch differences between the different differentiations. So we take the same iPSC, make it multiple times, and as you say, moon, sun, whatever, it influences it. But we also think that it is actually, of course, it's not perfect when we want to use it in the model for in the biohybrid, but it may add also to the biological variability. So we have a certain biological variability, maybe not a true biological N, but something that is going in the direction while controlling for the genotype. Mm-hmm.
Let me jump into that. If I have still, So that brings me to your proposition 4, where you say vitamin K should be pretty much given to everyone with the drinking water, whether they are sick or healthy.
Yes.
That's a pretty bold statement. Is this true for everyone? Because if you think about patients or people who have a genetic susceptibility for thrombotic disorders, that might be actually quite detrimental. That's why they actually invented warfarin. So do you think if you make that statement as a preventive medicine, that everybody should be either genotyped or have their blood worked up for coagulation factors?
In principle, that would be the right thing to do. I agree that, of course, not for everybody. Also, people taking warfarin, they should probably not take vitamin K, as it counteracts the intentions of their doctor. But I think for the vast majority of people, the statement holds.
Okay. Do I have one last question?
Yeah.
A very short one. Brings me to, I think it is, let me see, chapter 5. I stumbled across, when you use the serum of your patients and compare it to the healthy controls, you use pools of male and female donors.
Yes.
You gave the male and female donors for the healthy controls, but not for the chronic kidney disease. Do you know whether they were also reflecting a mixed pool, or was it only male or female patients?
That I don't know. For those donors, we don't know anything.
Okay.
Yes.
It might be worthwhile looking into gender effects. And with this, I would like to yield back to the prorector. Thank you.
Thank you, Professor Stoll. The opposition will be continued by Professor Hemmelder. He was also a member of the assessment committee and he's professor of nephrology at Maastricht University.
Dear candidate, I also want to congratulate you with your completion of your thesis, and also your supervisors I want to compliment with that. And I am a clinician, I work as a nephrologist, so I see a lot of people with reduced kidney function, and they all suffer from the effects of progressive vascular calcification. So I'm delighted that you put some insights in the mechanism of the vascular calcification, and I want to discuss with you on that because we cannot use yet the biohybrid assay in the clinic. And I hope, of course, that we can in the future use a kind of assay to detect in an early stage of kidney disease those patients who are at risk. So I'm very triggered on your chapter 5, in which you are testing 4 uremic retention molecules. And can you explain me why you choose these 4?
Yes.
Because in your table, you have perfectly shown that there are a lot of other retention molecules which are also maybe interesting. So why those 4?
Highly esteemed opponent, thank you very much for the question and also for partaking in the assessment of my thesis. Yes, those 4 were chosen. So originally what we wanted to do was differently. Originally we wanted to build a model that sorts all of the uremic toxins according to their chemical classification, so that we get clusters which resemble similar molecules. We pick a prototype, we measure it, we see effects, and we extrapolate the knowledge on the others. Because what I think is, now we added to the knowledge of 4 of them, that leaves still 100 untested. That of course, it adds more to the knowledge that has been before, but it's not as much as I wanted it to be. So the sorting of the molecules into chemical clusters did not work. They were just chemically so different that it didn't work, and also it neglected all the proteins because they wouldn't fit into a chemical sorting algorithm. Yes.
Yes, I understand that you had to do it with those 4, but if I look to those 4, there are 2 of them which have a pro-calcific profile and 2 are not.
Yes.
And were you surprised with that observation? That there are 2 who don't have that pro-calcific profile? Because when I look at the table and I look at differences between them, there are just a few differences and they all have a high score in your table. Why is this difference existing?
That is a very good question. So for the MGO, for example, we do know that it has different effects, whether it is added endogenously, exogenously, or produced endogenously. So it might be that difference. Also, this has just been done by us one time. Somebody else should repeat it in a different model at a different experiment somewhere else to confirm that. And that is actually true for most of the uremic toxins that are in the list, that most of the information that is in there has only been done once. And this is definitely a flaw in my table. So what I should have added is another layer of, okay, this has been done so many times, and then give it weight in the table as well, which I have not. I should have done that.
Okay, thank you. And if you look to those uremic retention molecules which did have an impact on the pro-calcific profile, then I want to discuss with you also figure 2 at page 94 in chapter 5, in which you describe dose response curves for paracresol sulfate.
Yes.
And you see higher doses, higher concentrations have a higher pro-calcific profile. How do those concentrations correlate with kidney function?
Well, there's inverse correlation. So reduced kidney function, at least for the paroxyl sulfate, is known that there is increased paroxyl sulfate with reduced kidney function, and with progressive kidney function, there's a progressive increase.
And where does it start to increase? At what level of kidney function?
At about 60, like a filtration of 60 ml per GFR.
And that's very early in the course of chronic kidney disease, because that's the start of chronic kidney disease.
Yes.
And then you already see an increase in dosage of concentrations of paracrystal sulfate.
Yes.
So that's the starting point of vascular calcification.
Well, the patients might have vascular calcification before that. It might just accelerate what's happening.
Yeah. Okay. And for me, it's very important that in the in part C of that figure of acrolein, you see that there is an on-off mechanism. With a low concentration, there's no effect, and with a high concentration, there is an effect. How do you explain that? Is there also a dose response between that?
That might be. I've actually not done that, because once we had the results of figure 8, then we looked at the clinical relevance and decided paroxyl sulfate is the one to go. And we just did that one experiment so we could show, okay, cell death related concentrations also trigger vascular calcification. And from that, we kind of concluded cell death is probably the mechanism. If there's a dose dependent cell death, we don't know. We didn't test that.
Okay. And would it be interesting to combine those to see if there is a synergistic effect? Because the mechanisms are different from each other.
Yeah, probably. And there probably would be an effect if we combine those two. So when we prime it with paroxyl sulfate and the cell is already stressed, it can't cope as well anymore with the second stressor. I believe that to be true. But also making these combination experiments, I think it's a bit of a rabbit hole, because where would you start and where would you end? That's why I did not do that.
Maybe you first have to adapt your table and to put in the value of the number of experiments which have been done, and you can recognize which kind of molecules are suitable to combine in those experiments, I would suggest. And I think it's very important. You used sera of hemodialysis patients in this study.
Yes.
Would you choose, if you do it again, would you choose that sera again if you would repeat those experiments, or do you use sera of other cohorts?
If we don't know whether our model works yet, then yes, because the CKD 5D serum has the strongest effect. So just to prove that the model works, yes, but doing a dose response with the different CKD stages, starting from maybe 2 to 5, is definitely an experiment we missed out on. Yes, so I would do that.
Okay. Is there some time yet?
1 minute.
1 minute. Then I go quickly to chapter 6, because that's also a very relevant chapter for the clinicians. When you look at 4 different cohorts, and 2 of those cohorts are CKD patients, can you explain why you choose for those 2 cohorts and not for other cohorts?
For which 2 calciphylaxis?
You have 1 cohort with hemodialysis patients, and you have 1 cohort with calcific uremic arteriolopathy,
yes,
which is a very rare and extremely high mortality disorder, which we don't see very often in the clinic. It's a very relevant that you had this cohort, but it's not a very representative cohort for the clinician. So why did you choose those cohorts? Because it's the outer spectrum of chronic kidney disease, and I would be more interested if we chase the storm to do it in a previous phase, that we can install preventive measures, you know?
That is true. So the calciphylaxis patients, they were indeed rare, but for us it was relevant to see whether or not we can still pick up differences between the hemodialysis and even one step further. And also them being rare is, in my opinion, maybe not a reason to exclude them, but rather to include them, because they have, if I remember correctly, a survival rate of less than a year when they're diagnosed with this. So if we can, for example, if we what we propose, develop the model further and then can predict which medication we can use to treat this specific sample, then it would be also valuable to know if our biohybrid system works with those samples. And for the hemodialysis patients, I must confess, when we did those experiments, we only had dialysis serum. Now we also have for other stages. And those are different experiments we will do in the future.
Yeah, it would be very interesting, I think, because that's the Leuven cohort you selected.
Exactly.
And they all were transplanted after that. That it would be interesting to see what happens after 1 year kidney transplantation with those measurements you make. But that will be maybe a next colleague of you will investigate that. Thank you, and I'll give the word back to the prorector.
Thank you very much, Professor Hemmelder. The opposition will be continued by Dr. Matic. She was also a member of the assessment committee, and she's associate professor of molecular medicine at the Karolinska Institute in Sweden.
Thank you very much, and thank you to the candidate and to the supervisors for inviting me to participate in person in this wonderful ceremony that I have only seen on Zoom previously, and it's definitely a different feeling to be present here. So I'm glad to have a chance to discuss with you. I happen to have a background in kidney disease from my own PhD, and then later I stepped more into the cardiovascular disease and vascular cell biology. But still, I think reading your thesis, I got to learn a lot of new things, and I think you, all together with this body of work, you bring a considerable new knowledge to the field. So my question to begin with is, going back a little bit into the etiology of both CKD and cardiovascular disease and commonalities, what is the prevalence, the epidemiological prevalence of CKD in cardiovascular disease, and if you know also the vice versa, what is your opinion, is the chicken or the egg in this story?
Highly esteemed opponent, thank you very much for the compliments and your question, and traveling all the way from Sweden, I much appreciate it. This is a very interesting question, and I don't have a good answer for that. What I know is that the prevalence for CKD is about 12%, like overall, and when we look at the cardiovascular disease, I'd say the prevalence is the same. So they pretty much all have, to some degree or to another degree, cardiovascular complications.
And your thesis specifically revolves around calcification as one of the commonalities, one of the common mechanisms. It is absolutely one of the most important mechanisms. Can you imagine, are there some other mechanisms that could be common, that could be driving chronic kidney disease in cardiovascular patients and vice versa?
Well, vascular calcification is already at the very late stage.
Exactly.
It starts, of course, earlier, and I think this feeds into the idea of early vascular aging. And I read a paper recently that beautifully put it as the arteries being a long-term memory of all stressors and risk factors that have been applied to your body, because the arteries don't forget, especially the elastin, for example. It's only produced when we're young, and then it has a lifetime or half-life of about 25 years, I believe, and after 50 years it's pretty much broken up and it won't be restored. So every stressor you apply will speed up the process and you will feel it later in life.
Okay. I think you're on the track with this answer to what I'm also thinking about. So definitely arteries as the connection between, and the vascular system as the connection between kidney and the heart. And one parameter that you did not include in your thesis, which probably it's difficult to get it from the patients, or maybe you don't get it routinely, and I know it happens to be a very unreliable parameter, and that is blood pressure. So do you think if you had it included that it would, give some weight to some of the prediction models that you have been doing in your chapter, I think 3 and 5?
Yes. Certainly it would, as it precedes vascular calcification and is at a very early stage of arterial stiffness, it will definitely add weight to the model. Yeah.
So then let me ask you some detailed, difficult questions about the role of smooth muscle cells in your study. You've done a lot of excellent cell culture work, and you have partially already answered what I was interested in, and that is how do you get these primary smooth muscle cells? So you explained you get them from organ donors and at aortic surgery, and there may be a chance that they are already predisposed to some form of calcification. I noticed that in all of your studies, you very carefully screen them for the expression of some contractility markers. But I'd like to ask you, do you think that the ones that you are testing are sufficient? You know, could there be changes that you don't capture with these markers that you are using?
Certainly, yeah. There are some that are more specific, like calponin or phosphorylated myosin light chain. There are others, like asthma, which are not specific to SMCs.
Yeah.
But overall, if I look at the vasculature, to pin down a certain cell type is almost impossible. As also the vascular smooth muscle cells, they vary in their phenotype a lot.
Yeah.
So I think what we define as a vascular smooth muscle cell is just an expression of a certain protein profile.
True. Do you know of any sort of more sensitive markers that become modulated immediately when smooth muscle cells go into some kind of a change or a transition?
Well, yesterday I heard they will change the shape of their nucleus.
Yeah,
when they go from elongated to less elongated.
Yes, that's one possible symptom of a change. Yes. Okay. So then another question is about the biohybrid assay. I think here you have done an excellent job, really, which still falls into what I would call a proof of concept study, but indeed an excellent job with putting together something that does have a clinical potential. And my question is actually related to that. So you've done 2 chapters about this assay. There's a lot of things that you have tested here on a smaller number of patients. So I want to ask you, if somebody tells you, now start to do actually something that would lead you to the clinic, to the application of this assay in the clinic, how would you design that study for the purpose of clinical application? What do you think is missing from the 2 studies that you've already done?
Automation is missing if you want to bring it.
Okay, yeah. Maybe that's not the first thing.
We need to downscale it when we want to do more patients, because in the setup that we have now, we can't do as many samples as we need.
Yeah, absolutely, yes.
Yeah.
Which is linked to the automation then, right?
Well, I mean, we can also do it manually in 96 ball plates.
Yeah.
Yes.
But the scaling, what kind of possibilities would that give you, which you didn't have at the moment when you were doing the study, because you've used, I think it was around 10 patients per category.
Yes, roughly. Yeah, roughly that number of patients. That's what I meant. I'm sorry, I don't know.
So you have already, even with these few patients, you have included some clinical information, right? But the scaling would allow you to include much more clinical information. So for example, if I am a clinician, which I'm not, I would like to know what kind of specific effects happen with the plasma from diabetes patients. Does that contribute to the calcification?
Yeah.
And you didn't have the power to do it at that moment, but is that something that needs to be assessed before the assay goes into clinical application?
Yes, for sure. I mean, especially the chronic kidney disease patients have a lot of comorbidities which we can't control for at the moment.
Exactly. That's exactly what I was getting at.
Yeah.
Do we have time for more questions or no? Yeah. Thank you.
Thank you very much, Dr. Matic. The opposition will be continued by Dr. Knowles, also a member of the assessment committee, and she is Associate Professor of Molecular Cardiovascular Research at the University of Aachen.
Dear candidate, also from my side, first of all, congratulations with your thesis. It's really a huge amount of work. And not only that, you also managed to really nicely write this together in a great thesis and also gave a great presentation. But nevertheless, I still have to fire some questions. So I would like to start with intimal calcification. So you mainly focused on the medial calcification, but you also mentioned in your introduction that there is also intimal calcification and plaques. And you said that there are some differences between intimal and medial calcification. So my first question is, what are main differences or maybe also main similarities between both?
Highly esteemed opponent, thank you very much for the question and coming all the way from Aachen. And also thanks at that point for leading our consortium. I had a really good time and it added a lot to my PhD. Thank you for that. And yes, regarding your questions, I think they are both fundamentally different and fundamentally the same. The processes that are common are cell death, oxidative stress, inflammation. As for inflammation, I'd say it is a more local inflammation in the intimal calcification, whereas the more systemic inflammation drives the vascular and the medial calcification. uremic retention molecule effects contribute probably to both. What is a bit more specific to medial calcification, I would say, is I forgot the word, senescence, and loss of inhibitors is perhaps a bit more specific to medial calcification, because I think you can't talk about loss of inhibitors in atherosclerosis when the environment that is created is not natural in the body, so it naturally doesn't have inhibitors. It's just a new created environment. And I think those are differences between both.
Yeah, okay, thank you. So yeah, you already mentioned then some processes that are important, like cell apoptosis, inflammation. Which of these do you think is actually most important? And so also cell differentiation or smooth cell dedifferentiation was mentioned. Do you need all of them, or is there one which is most important? And if you have that process, like, I don't know, apoptosis, you will get calcification, or do you need a combination of processes?
You need a combination of processes. It's never one thing that happens alone. It's also because these processes, they induce each other. So you cannot usually not have one without introducing at least one other, I would say. So they're definitely working all together.
Mm-hmm. Yeah, okay. And so yeah, processes of inflammation and also reactive oxygen species, which you also studied in your thesis, you see them not only in smooth muscle cells, also in other cells. So I always wonder, why is it that the smooth muscle cells contribute mainly to calcification, and is it also other cells, and is it through the same processes? For example, if I just induce apoptosis or inflammation, would I then also get calcification?
Yes. So the smooth muscle cell is, in my opinion, if I had to nominate the cell for Academy Award Best Actor Vascular Calcification, it would be the smooth muscle cell, definitely. That'd be cells nominated for Best Supporting Actor. For example, macrophages, endothelial cells, when we talk about intimal calcification. Mm-hmm. When we especially talk about chronic kidney disease calcification, I would put all my money on smooth muscle cells, because when we, for example, look at children with CKD, they also at the later stage develop calcification, and it is usually like specifically medial calcification and not intimal calcification. So I think medial calcification is in that case even more specific, and that mostly or exclusively involves smooth muscle cells. For the intima, endothelial cells definitely play a role, as they're stressed also by uremic toxins, and the first cells involved into the development of atherosclerosis. Macrophages are then at the second stage recruited, pulled into the plug and make it develop. But what I've read is about atherosclerosis, most cells are smooth muscle cells, or at least of smooth muscle cell origin, although they might adapt a different profile of protein expression later. And especially around the areas which show calcification, it's smooth muscle cell origin cells that are most prevalent.
Okay, thank you. And yeah, maybe then going one step further in this inflammation. So inflammation was one of the processes that comes back, and also in your findings with paracarsyl sulfate, oxidative stress. Do you think that interfering, like in clinical trials, with inflammation, oxidative stress, would that also help to reduce calcification?
Studies have been done on trying that. None has yet been successful, as far as I'm aware, to do it in a broad sense. So the non-steroidal inflammatory inhibitors like COX-2 inhibitors, for example, have been tried. Statins are in use a lot, and despite being anti-inflammatory, drive vascular calcification. So just using a anti-inflammatory approach alone is probably not enough. Neither is trying to inhibit oxidative stress development. It probably needs and that's what we argue also, that we need to understand the processes in more detail so we can have more targeted approaches.
Yeah, okay, thank you. Yeah, so let's then go into these specific processes, and I move to chapter 5 now, where you study the effect of paracarsyl sulfate on vascular calcification. You mentioned, for example, that paracarsyl sulfate is inducing extracellular particles, several other processes like ROS and also RAGE signaling. What about these extracellular particles? Is that somehow linked to your oxidative stress or your RAGE signaling, or did you test whether then, okay, you see them increase? Do they also play a role? Did you examine that?
So far, we can't pinpoint a specific role to them. We believe they do play a role. That's also why we tested, for example, in the lower two corners, you see the GRP78 and the PPERG, which are endothelial extra ER stress markers. which are also linked to extracellular vesicular release, and that's why we also checked for NOx5, which we've shown before it is involved in the vesicular release. We can't show at that time points that we looked at that they are directly causally involved in the vesicle secretion. And also what we don't know is the vesicular particles, actually. We don't know their composition. So far we only know it's more of them. But we don't know, do they actually contribute? Don't they contribute? Because they can also have different effects depending on the surroundings. So we don't know that. For now, it's a standalone finding that there's more.
Yeah, okay, thank you. Is there time? No time. Okay, thank you very much.
Thank you.
Thank you, Dr. Knowles. It's now time for the last opponent. It's Dr. Donners. She's Associate Professor of Pathology at Maastricht University.
Thank you very much. Dear candidates, I also would like to congratulate you, but also your supervisory team for a very nice piece of work. I really enjoyed reading your thesis, and many questions came up. Some of them have already briefly touched upon, but I was particularly intrigued by your title, Chasing the Perfect Storm. What was that perfect storm for you, and did you in the end find it and beat it? Can you briefly explain?
Yes, highly esteemed opponent. Thank you very much for your compliments and your question and for coming today to spend the time. So the perfect storm is everything that develops, for me it was everything that develops in chronic kidney disease and then drives vascular calcification, so all the factors together. And so although I say I was chasing the perfect storm, I only chased maybe one little cloud in the storm, which are the uremic toxins. All the other factors in the storm we neglected. Yeah, and I'd say maybe we came in the shadow of the cloud, but the cloud is still to be chased.
Yeah. So I'm talking about these uremic retention molecules, the ones that you used in your chapter 5 to look at the impact on smooth muscle cells. I couldn't really find where you got them from. Were they commercially available, or did you isolate them? Because usually they are protein bound in patient serum, right? So
exactly.
Was that also the case for the uremic retention molecules that you used in your screening?
For one, yes. For the paroxyl sulfate, that's true. And we bought it just commercially. You can buy it as a salt.
Yeah. But then I assume they're not bound to any proteins, and I can imagine that would really impact their function.
They're, of course, as long as they're not soluble or in contact with proteins, they're not bound. It has described that albumin has 2 binding sites for it, and we have albumin in our system as we add serum. So there might be a binding happening while we add the crucyl sulfate to our cell culture conditions. As to the fact whether or not the free or the bound fraction does the effects, this is still also under debate. Some people say it's only the free fraction, but in my experience, I've never seen proof of somebody actually showing that's true. Yeah.
Okay. Then you talk a lot in your thesis about using the biohybrid system. I assume with the stem cell derived smooth muscle cells in personalized medicine. And I was wondering how you envision that. Which people would you like to screen with this system? How would you apply it?
Yes. Firstly, of course, while we built it, I'd say high-risk patients, and then going back to what we heard earlier, starting at CKD stage 3, and trying to identify high-risk patients, which maybe need more attention. Or we can look at which profiles specifically are in the serum of a certain patient, while we use their own cells to put the serum on. So we have a perfect match.
Yeah, but when you talk about patients, they often present in the clinic when they already have calcification. So wouldn't it be better to check in your biohybrid system drugs that could reverse the calcification? And shouldn't you then, instead of the smooth muscle cells, which you consider to be the most important one, and I assume for initial screening for the propensity of calcification, that's true, but if you think about patients, treating patients, reversing calcification, shouldn't you look at macrophages, maybe even osteoclasts that could resorb the calcification?
Yes, that's a very interesting idea. Of course, I've never heard of a drug that is supposed to be given to a patient to regress calcification. Also, I've never seen a paper where they actually regress calcification in vivo. If you do it in vitro, you of course have other mechanisms to do that. Driving a macrophage towards osteoclast phenotype would of course be interesting, but how do you direct that macrophage to the calcification and not to the bone or to the teeth? Doing that in vitro sounds fun, but doing it in vivo, I don't see how how could we specifically direct it to the place where it needs to be.
But you already talked about inflammation being contributing to the calcification, but then the macrophages are there, and you could try to switch them to more resolving phenotype, right?
That's an interesting idea. If that works, that sounds perfect.
I think there are plenty of opportunities for further research, right? Then another question, in your impact section, you talked about all the advantages of the biohybrid, and I can only underscore the importance of this system, and I can acknowledge they have many advantages, but I can also think of some disadvantages. Could you think of any limitations of this screening system?
As it is now, the variation is still way too high. So we can't use it at the moment to do this in time for a patient. When we want to do iPSCs, take blood, make iPSCs, make smooth muscle cells, it takes about a year. So that's not feasible. So at the moment, we can't really personalize it. That's definitely one thing. And then...
Thank you very much. Maybe you can finalize your answer later on. Nikolas Rapp, the time appointed for defending your thesis has passed. The degree committee will now withdraw to discuss the quality of your thesis and your defense, and I request that you and your company await the results of our deliberations in this room and wait till we return.
Thank you.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
Long road to the south side. 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and that. All your time, 'cause I'll go the extra mile. Long road to the south side, 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and that. On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose, bad branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off. Take the mileage, traveling so far from home. Gathering knowledge, other connections. I put the time in to get everything I ever hoped for. It's a challenge and we all in. It's a better future that we go for. Even though these nights seem long, I know that I am never on my own. Know you won the battle once you've grown. South side, 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home. If it's questions that you've got, go the extra mile and not. Long road to the south side, 10 miles in my rearview mirror. I know what it felt like. My goals only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and not. Years can't fly by now, don't waste all your time. 'Cause I'll go the extra mile.
I'll faint.
Oh, there you go.
I don't know.
Nikolas Rapp. The degree committee here present has assessed the quality of your thesis and your defense. In view of its positive verdict and taking into account your previous qualifications, the degree committee has decided to grant you the degree of doctor. Professor Schurgers is authorized to confer upon you this academic distinction in accordance with Dutch university custom. I invite your supervisor to now take the floor.
Before we do so, Nico, you have to promise me something. Do you promise to work in accordance with the principles of scientific integrity at all times? To be careful and honest, transparent, independent, and responsible?
I promise.
By the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Nikolas Andreas Rapp, the degree of doctor and grant you all rights attached by custom and law. As evidence of this, I now present you with the degree certificate signed by the rector, the secretary, and all the other members of the committee and affixed with the official seal of our university. Dear Dr. Rapp, dear Nikolas, dear Nico, from this place I'm the first one to congratulate you with this great achievement. In these congratulations, I want to also include your family, here well presented today, congratulations, and also, of course, your girlfriend, Sophie. I also want to thank, before I go over to you, the members of the assessment committee and also the members here present today, and then especially also for Dr. Lubika Matic for coming all the way from Stockholm. Thank you very much for this. Dear Nico, our story goes back to 2018. After the ITN IntriCare, where we had a collaboration between Maastricht and Aachen, it was now the other way around. It was the Aachen-Maastricht connection that secured another ITN called Caresian. And that was led by our Aachen partners, Professor Jankowski and Dr. Heidi Nuels here present today. Thank you, Heidi, also for being here. So I could appoint a new PhD student for this ESR 7 position. In the final round, we selected 3 candidates for a personal interview in Maastricht. And to be honest, our team had already put all the money on this German guy. And this German guy was not Nico. This guy was working at Harvard University as a master's student. However, we wanted to give all the 3 candidates a fair chance, and all were invited to Maastricht. And Nico was invited because his application stood out. A well-written motivation letter and a somewhat interesting CV with the title, On the Trails of Marco Polo, to Travel and Explore. And with that, there was a photograph of yourself that looked to me as a photo of a young high school boy. Also, your recommendation letters sounded very promising. Goes from very well-written thesis, graded with the highest remarks, and very organized, reliable, excellent abilities to understand and explain experimental procedures and scientific results. Continued with the second recommendation letter. Nikolas aims to be the best at his craft, constantly learning, working long hours, attending every seminar and reading every journal paper. He can own his skills. He has curiosity and willingness to constantly improve and adapt to new techniques. So that was very, very promising. So after having listened to the presentations and discussed with all three candidates their work in detail, it was clear to the whole assessment committee that there was only one clear number one, and that was you, Nico. Only a few months later, precisely the 12th of June 2018, you started in our Maastricht finest department, the Department of Biochemistry. This surface dude with ponytail, in German Zopf, also termed by my wife as the ideal son-in-law, was soon called Handsome Nico in our group, because we had suddenly two Nicos. Your project within the Parisian consortium was ESR7 and entitled Effect of Uremia on Vascular Calcification in the Cardiorenal Syndrome Pathology. And as with most PhD students working in my PhD group, I give you some freedom in the beginning. This to promote the to find your own path and intrinsic motivation within the boundaries of the project. And in the beginning, I had no doubts about Nico. But at a certain moment, I started wondering how the project was going. Nico worked long days, was always friendly, a nice guy, saying hello, but never asked help or advice. During our group meeting, he presented his work in a very detailed and structured manner, in very good English and very academic. And before I could ask Nico about this, where all the results will be, after a half year or so, he simply presented his first paper to me and said, "Here is a more or less ready to publish story." And so, Nico, you really stood out in that. You also quickly felt home in Maastricht, making a lot of new friends. Not in the last place at work with Cengiz and Greg. You made a lot of city trips together, having fun. And maybe good, I don't know all the ins and outs, what happened during these travels. This friendship also led to collaboration and resulted in several shared publications. And this is how it should be, Nico. And indeed, it was a great pleasure having you in Maastricht, since you enriched our group with mature, friendliness, hard-working mentality, team player mentality, calmness, warm and positive personality. But for me, Nico had also some mystery around him, because I rarely saw him for the discussions. He was very mature and worked on his own. And in the beginning, I ascribed this very much to his independence nature. But after some 1.5 years, Nico came to my office and confronted me with the fact that he felt alone in science, not sufficiently guided in his project, and that he was losing fun in academia. This was quite a shock for me, Nico, but also a wake-up call. I realized that during this meeting, the teacher learned from the student. Without going into the details, we had a very good open meeting and many more that followed. After that, your work flourished even more. The result can be seen from the thesis today and the nice doctorate. And for me, you helped me reflecting to my work as well, therefore doing my work better and getting a better supervisor. So thank you really for that, Nico. And reading your acknowledgement of the thesis, this meeting clearly worked out for our relationship and friendship. So thank you for that. I truly believe you found some fun and love back in doing science. For sure you have, as the Germans would say, das gewisses etwas. However, the love for Sophie is way bigger. And I believe there is no job or science that can beat that. You met Sophie here in Maastricht and now moved together to Vienna. You found there a job in Vienna at Roche, the company that you left for your PhD to come to Maastricht. One can say it makes the circle round again. Nico, I wish you all the best for the future, and I'm pretty sure whatever you will do in later life, you will succeed. For now, the thesis defense is over. The stress, nervosity are gone. You can go back to relaxing mode and enjoy this and celebrate this fantastic achievement and moment. Dear Nico, I hope, but I'm also pretty sure that we stay in touch. Nico, es geht dir gut, lebe wohl und hoffentlich auf Wiedersehen.
Dear Dr. Rapp, also on behalf of Maastricht University, I would like to congratulate you with the degree you have obtained today. You did very well in your defense. The committee was quite convinced that you deserved the degree. So I hope you also yourself look back on this ceremony with satisfaction, also on your whole time in Maastricht, of course. And I would also like to congratulate your family, your partner, friends, colleagues, without whom you probably wouldn't be here. Colleagues are always very important. I'd like to congratulate your supervisors, and I'd like to thank all the members of the degree committee here present for their contribution to this ceremony. It's very much appreciated by Maastricht University. And it's now time to celebrate. So I close this academic ceremony. And now it's time for other things, I guess.
Thank you. Thank you very much.